Thank you guys for joining me this morning. I want to share a couple of things today with you. First, a little bit about general ideas of soil health, and then since I am a soil microbiologist, as Mongi mentioned, a little bit about why microbes are important when we're thinking about soil health, and then share some information and data from some projects that we've been doing with my program and colleagues about some of the methods that we've been looking at for trying to improve soil health, and in particular, that soil organic matter component. So first off, I think most of you guys, if you've heard me speak before, have probably seen some of this information. But I think it's always important to keep in mind when we're talking about this idea of soil health. And first of all, what is it? How do we define it? And so there we go. One of the main definitions that's often used is the one from the USDA NRCS about the continued capacity of a soil to function as a vital living ecosystem that sustains plants, animals, and humans. And for those of us that have been thinking about this for a while, this certainly seems similar to this idea of soil quality or soil fertility. And for the most part, it really is the same general concept, with one big exception in that soil health is really also including and having a bigger focus on soil biota, on microorganisms, which previously, when we were thinking about the idea and the concept of soil health, soil quality and soil fertility, most of the focus was more on that chemical, physical properties of soil. And now over the number of years, we've realized that soil biology plays a really big role. And so that's where this idea of soil health comes into play. But despite all this talk about soil health, I think it's still kind of a nebulous concept. And so what I think is really important to keep in mind when we're talking about this idea of soil health is to really think about how soil health plays into our understanding of the overall resilience of an agricultural system. It's what is that function of having a healthy soil? What are we really looking at it to do for us? And I think that's what we're really thinking about in our agricultural systems in particular is resilience. And so when I'm talking about resilience in agricultural systems, I'm talking about the ability to bounce back from changes in water or nutrient availability, climatic extremes, which obviously we see here in Florida, and pests and diseases. And so having this idea of a healthy soil can potentially deal with some of these issues and provide that resilience. So where do soil microorganisms come into play with this? Well, it turns out that soil microbes are actually controlling some of these same aspects of resiliency. For example, there are microorganisms that are involved in the availability of nutrients. And as I mentioned earlier, having one of the concepts or one of the ideas of having a resilient agroecosystem is having these nutrients being available when they're needed. Certainly, soil microorganisms are causing some diseases, but there are also a huge number of beneficial microorganisms that are in the soil. And that's something that we also want to think about when we're thinking about having a healthy soil and having a resilient soil. There are microorganisms that are known to stimulate root growth. Of course, in citrus, this is of particular interest given HLB and the impact on the root systems. And so microorganisms that are present in the soil or increasing the number of those microorganisms in the soil that could enhance root growth could be potentially beneficial. And finally, one of the other big aspects of thinking about soil health and even the role of microorganisms is this role of aggregation in the soil. And so there are a number of microorganisms that release various compounds that can help aggregate soil, which in turn can help with nutrient and water availability and having, again, a potentially more resilient system. And as you know, these are all potentially important things in particular because of our Florida soils. And so as you all know, we have basically beach sand that we're growing our citrus on. We have low organic matter, very low nutrient availability. And so that makes growing crops very challenging, and it makes altering the soil microorganisms a little bit challenging. Keep clicking on the wrong place. So as Mongi mentioned, one of the key ways, though, of improving our soil health and potentially boosting this resiliency of our agricultural systems is increasing the soil organic matter. Since we have so little in our Florida soils, any little bit could potentially assist and provide resources for making our systems more resilient. So organic matter is really the decomposition of plant material, animal material, and even microbes. And so this is a sort of a circular system in that microorganisms are going to help decompose this organic matter, which provides food and resources for these microorganisms. And as these microorganisms die, they also provide more organic matter. And so this then helps build that organic matter into the soil. It's a slightly more complicated process than that. And again, we could probably do a whole talk about that. But again, as that organic matter is formed, it helps stimulate the microbial community, which can then decompose even more and cycle this whole system through. And so having that soil organic matter, one of the key things is that it is going to provide resources for microorganisms. And so having more of those microorganisms increases your chances that you're getting microorganisms with the benefits that will help your system. So how do we improve our soil health here in Florida? How do we improve our organic matter here in Florida? There are a couple of different ways, and none of these are new methods. These have all been methods that have been used for, thousands of years since we've been doing agriculture. They've just come in and out of favor for various reasons. And so what I'm going to talk about today is cover crops, compost, and humic or fulvic acids. I'm going to be focusing on humic acids. And so these are all methods that have multiple potential benefits, but one of the key benefits is, and that we're often focused on, is this generation of soil organic matter and the alteration of that soil microbiome, the microorganisms in the soil. So I'm going to start with cover crops. As you probably know, cover crops, their whole existence, by definition, they are planted to benefit soil health. They're not harvested for profit. They've become increasingly common for row crops and some vegetable production. They've been used historically in perennial crops like citrus and certainly are used by citrus growers and other tree crops, but they're becoming more common, I think, for tree crops than they have been in the past. And in these annual systems where they have been common, they're often combined with conservation or no-till management practices. So there are numerous potential benefits of cover crops. One of the big ones is that they can provide nitrogen if you're planting legumes, or they can potentially scavenge nitrogen from a previous crop. This is more the case in annual production than in citrus trees. One of the big benefits is that they can reduce weeds. They can outcompete weeds. They can reduce soil erosion. They can reduce soil compaction. They can also increase soil moisture, and the big one, increase soil organic matter. In fact, increasing soil organic matter is one of the reasons why you might have increased soil moisture. So a lot of these things kind of go together. So I've done a few presentations, and there's been some discussion in the past about what are some of the practical considerations for planting cover crops. And so I'm just going to go over a few things briefly. I will mention that we have an EDIS document that is under review. Hopefully it will go through the review process very quickly, and that will be available, which has some of this information in it as well. So the first thing is, where are you going to plant your cover crops? And so obviously we've got a couple of different, or at least two different ways that we plant citrus here in Florida. And so in the south here, in the flatwoods, we do this swale system, bed and swale system. And so we've been primarily planting our cover crops just on the raised bed row middles. This is a picture of a trial that we have here at the research center with different treatments. I do know some growers that have been experimenting with planting cover crops in the swale, and I'm super curious about that. And, I think that potentially has a lot of benefits too. There are just some logistical challenges that you have to think about with that as well. Up on the flatwoods, of course, sorry, up on the ridge, of course, then we don't have that swale system. And so then you can plant cover crops on both sides of the tree, which also has some additional benefits that we might not see by only planting them on the raised bed middle down in the flatwoods. Again, a lot of this is just practical considerations that you have to think about with your grove and how you're operating. All of the research trials that I've been involved in have only been involved in planting cover crops on the row middles and still leaving the area underneath the canopy clean. We've not done any trunk to trunk planting. I'm certainly curious about it. I think there are some pros and cons to it, and I'm happy to discuss that, but I don't have any research data on it. We're still leaving an area, even though it looks like there's not much in this picture here on the left. There is still a clean space underneath those trees. Those are just really big cover crops that summer. So the other thing that we want to think about and that we've been doing in our research trials is doing 2 different plantings of annual cover crops each year. So one at the beginning of the summer to try and capture that excuse me, that the beginning of the rainy season, and another one at the end of the rainy season to capture the end of the rainy season, the rain right before. This is some older precipitation data, but you can see, we have as everybody knows here, we have all of our rain more or less in the summer, and so we're trying to capture that. The idea is that we're not fertilizing and we're not irrigating these cover crops. We're just letting natural precipitation take care of them. And so getting that timing right can be a little bit tricky. Summer planting, we often try and get somewhere in June and generally don't have too many issues then. Winter planting is the one that I find to be most challenging, is trying to make sure that you're letting your summer cover crops grow as long as they can. We like to let them grow and flower and generate seeds if possible to generate a seed bank, and then mow them and then plant our winter cover crops while we're still getting a couple of rainstorms. Then the big question is always about what to plant. And again, this information is available or will be available in an EDIS document. I think it's also been published in Citrus Industry Magazine a few times. I'm also happy to make this available through email. But these are some of the different species that we've used in some of our trials. We have tried a whole number of other things. We have certainly tried a number of clovers, and we haven't had much success with them. We have not tried any other perennial cover crops like perennial peanut, primarily because they take a really long time to get established. And if we're trying to generate some data relatively quickly, that makes things a little complicated. But these other ones we have had good success with. In particular, I would say sun hemp has always been done very well. We actually plant sun hemp both in the summer and in the winter because it is one of the few legumes that still seems to be able to grow, at least down south here, during the winter. And I would say that daikon radish has been the big hit in the winter. That always seems to do really, really well. So what happens when we do all of this? Let's talk about some data. So one of the first things, like I mentioned, is that what we're really interested in is seeing how this impacts soil organic matter. And so this was a trial we did a number of years ago in a commercial citrus grove where we planted cover crops. We had 2 different treatments. We had a cover crop mixture with legumes and non-legumes. That's the green bar, excuse me. And we also had a mixture that only had non-legumes, and that's the orange bar. Our grower standard control is in blue, and that was basically just left to whatever weeds were naturally growing in the row middle. And so we can see on the y-axis here is the percentage of soil organic matter. And this trial, actually, that soil, that grove, had pretty decent organic matter for South Florida to start with. At 3%, that's pretty high. But after a year of growing the cover crops, we saw a significant increase in that soil organic matter. And that more or less maintained that difference for about 2.5, 3 years, and then the trial finished. So we were able to see increases in organic matter. I will mention we have another study that's going on right now with one of the postdocs in my lab, and we're finding that how deep you collect your soil really makes a difference. Not too surprisingly, but we'll have data on it, on whether you see changes in your soil organic matter. This seems kind of like a no-duh concept, but when we're measuring things for science, there are sort of some standard depths that are used. And it turns out that you might get different results if you collect your sample from 0 to 10 centimeters versus 0 to 15 centimeters. That 5 centimeters can make a huge difference in how this organic matter is distributed in the soil, and therefore whether you're seeing statistical differences. And so that's something that we're working on and confirming. But that's also something to keep in mind when you're collecting measurements of if you're trying this as well. The other really interesting thing, and I think, again, as a soil microbiologist, there's sort of two components to this. There's one, building that soil organic matter, and two, are we seeing changes in those soil microorganisms? And so, yes, we are seeing changes in what bacteria and what fungi are present in these soils when we're planting cover crops. That said, we don't always see these changes. And so this was a study that we did a few years ago where we had 2 different citrus groves. Both were in the general Immokalee area, so similar climate, general similar conditions. They were both managed by the same company, but one had a slightly different organic matter in their soil to start with than the other, and that made a big difference. And so what I'm showing you here on both of these charts is where each point represents the total bacterial or fungal community composition of one sample. So if the points, shapes, whatever, are really close to each other, that means they have a very similar community composition to each other. If they're far apart, totally different. And so we've got a couple of things here. So first off are these triangles and the little diamond shapes. And so these are this is the bacteria community composition of samples at our location B at the beginning and the end of the trial. And so the colors are the different treatments. And you can see there's no real pattern to the colors. There is a pattern in that the diamonds are all clustered together and the triangles are different than the diamonds. All that means is that the bacteria composition was different at beginning of the year and after 3 years, but the treatments, the cover crops, had no impact at that grove. However, same with the fungi. However, at our second grove, at our location A, we do see differences based on the cover crops. And so here, all of the points that are clustered in that big orange circle that I show was what the bacteria communities looked like at the beginning of the trial. So they were all very similar. And at the end of the trial, we see that the red and green ones, or those that were treated with cover crops, have a different bacterial community than the start. So we are seeing changes, or we can see changes due to cover crops on the bacterial community composition and with the fungal community composition. But it really depends on how well those cover crops are growing. And so in that first location, they really didn't grow that well. And again, seems like a no-duh point, but if they don't grow, we don't see that change in the soil organic matter, we're not going to see that change in the soil microbial community. Something that we're looking at a little bit more closely now with a newer study, and hopefully I'll have more information on this for you in a future talk, is not just who's changing, but what are these organisms doing in the soil? Because it's great that we might have differences, but what's really important is what they might actually be doing. We've played around with this a little bit in the past, and so we have ways of predicting what the functions might be of the bacteria in the soil. And so when we do that, we do see, so the orange and the green are, again, the cover crop mixtures. The green has the legumes, the orange doesn't have legumes. And so the darker shades are what things look like after 3 years of these treatments. And so we can see that there are increases in bacteria that have a lot of roles in carbon metabolism when we're planting cover crops. And we also see big increases in microorganisms that have roles in nitrogen metabolism when we plant cover crops. There's a whole bunch of other different functions that seem to be increasing or potentially increasing when we're planting cover crops. But these are all potential functions. And so what we're doing right now in my program is trying to isolate some of these organisms and determine what they're actually doing, and not just what they might be doing. And so that's one of the things that we really want to look at and say, how are these cover crops actually helping and changing some of these characteristics of the soil when we are adding soil organic matter in cover crops? So one other big question that I sometimes get asked is, all right, well, you're planting your cover crops in the row middles and they're not right next to the tree. Are they still actually changing things around the tree? And that's a very, very valid question. It's actually very complicated to try and answer in a grove, because if you try and collect samples of citrus roots that are planted in the middle of all these cover crops, that's hard to tease apart. So we did a greenhouse trial, and this is a trial that is being conducted by PhD candidate student in my lab, Emma Dawson, where we took cover crop soil from these cover crop trials and brought it back to the greenhouse, and we planted citrus trees into that cover crop soil. We also took back the control soil that didn't have cover crops. And so we looked at what was the bacterial community in the bulk soil and that soil around the roots, and what was the bacterial community on the rhizosphere soil, which is the soil that's right next to those citrus roots. And we saw that there was a big difference. And so this is looking at what we call the core microbiome. But the top here is showing the cover crop soil, is showing the bulk soil. And so that's sort of the general soil in the pot. And so each color is representing a different bacterial genera. And then the bottom is the rhizosphere soil. And that's the soil that's right next to the roots. And that's the soil that's going to have, and the organisms that are going to have the most direct contact with your citrus tree. And so we're seeing a very, very big difference in the bacteria that are surrounding that citrus root, whether it was planted in the control non-cover crop soil or planted in the cover crop soil. And in particular, what's really interesting is that these genera, Bacillus and Nitrospira, that are around those citrus roots that were planted in cover crop soils have potentially, again, potentially, we don't know for 100% certain, but certainly relatives of those genera are known to have benefits to plant growth. Bacillus has a wide range of potential benefits, including improving root growth and nutrient availability. Nitrospira are involved in the component of the nitrogen cycle, and so could be making nitrogen a little bit more available. So the citrus trees, when their roots are encountering the soil with the cover crops, which they certainly do reach that far, they are seeing different microorganisms, and they are recruiting different microorganisms from that bulk soil that has been planted with cover crops. So one last thing I do want to mention here is, actually, let's see, there we go. So real quick, the take home on cover crops. First off, we certainly have seen that cover crops can change the soil microbiome, and they can change what we think are the functions of these microorganisms in the soil. And most of that is related to this change in some of the soil organic matter. But the magnitude of these changes really can depend on the cover crop mixture. We see a lot of bigger differences, more differences when we're planting mixtures that include legumes. And it can depend on time. We've also seen that, yes, cover crops can impact the organisms that are growing around a citrus root, even if you're planting your cover crops in the row middle. But I think one of the big things is that we still have not seen significant changes to the tree. We haven't seen major changes in yield or other aspects of the tree. with cover crops. A lot of this has to do with how our trials have been conducted. For better or for worse, our trials we started with trees that were very, very old, 30 plus years old. And that's a little tricky to see quick changes in a tree that old. Another trial that we were hoping to let go a little longer ended because, as you know, as everybody's dealing with, we're having groves that are shutting down, which is sad and unfortunate for many, many reasons. We do have a trial that just started about 2 years ago here at our research center with brand new, newly planted trees that I showed a picture of earlier, and I'll talk a little bit more about in a minute. And we're hoping that because those trees are very young, we might be able to see changes a little faster in the tree. But again, that trial is just getting started. We also still have a lot of questions about some of the nutrient release and availability with these cover crops, and we're working on a project with a colleague about that, as well as this idea of carbon sequestration. So there's still plenty of research to do and many questions still to try and address with the use and benefits of cover crops in citrus. But cover crops are not the only way to potentially change soil organic matter and soil microbial communities. And certainly compost is, again, not a new way. And I'm sure many of you guys have tried this at various points or are using it right now. And you're probably aware that there are a lot of benefits and challenges with using compost. In general, in Florida, it's pretty available. It's not terribly difficult to apply, depending on a couple of key points. And it can be a nice nutrient source, again, depending on some of the factors that you're really looking at. However, it's not without its challenges. Depending on how big your grove is, it could be pretty expensive. There are no, I think, firm guidelines on application frequency, which of course could alter your cost. Again, depending on how much you need, maybe availability is a difficulty. There certainly can be some variability between compost companies and the material that you're getting, and it can potentially be a source of weed seeds. And so those are all things to keep in mind when thinking about using compost. And so my colleague, Dr. Uta Albrecht, as well as Dr. Ramdas Kanasari and I we're working on a few field trials. One that I want to talk about first is one that was done in a commercial citrus grove that, again, is unfortunately no longer with us, where we had 4 different rootstocks. And we applied either a compost or we had a no compost treatment to these trees. And we applied the compost twice a year at about a 12.4 ton per hectare rate. And we collected samples, and Dr. Albrecht did a large number of things with these as well. But one of the things that we were particularly interested in was, again, what is anything happening with the soil microbial community when we're adding this compost? I want to point out this picture it does actually show the compost underneath some of these trees, but it's when you think 12.4 tons per hectare is a lot. We were sort of amazed. Maybe this was me being a little naive. When you apply this, it's a really light dusting. We actually had it applied incorrectly to one row that was supposed to not get compost, and we had some graduate students take a leaf blower and were able to blow that compost right off. It's that fine a layer. And so I'll be honest, I was skeptical as to whether this was going to do anything to the soil microorganisms. But long story short, yes, it did change the bacteria that were surrounding the roots of these trees. However, it really depended on the rootstock. And so there were bigger differences in that bacterial community and that microbial community based on what type of rootstock was there. And so in this diagram, the colors represent the different rootstocks. All of the squares here are the control treatments. So all the control treatments, for the most part, there was really no difference in the microorganisms that are around the roots without compost, regardless of the rootstock. But then once we add compost, we see differences in that microbial community around those roots based on the rootstock. So there are different microbes around the roots of US 897 in green compared to around the roots of US 812 in red. And so that was really interesting, and that we were seeing this rootstock effect in response to compost. And this is a little bit correlated as well with some of the changes in the nutrients in the soil and in the roots. And so this was, again, collaborating with Dr. Albrecht and some of the work that her lab has been doing. And we see that there is a really strong positive correlation in changes in the microorganisms with changes in magnesium, same with zinc and manganese. And so these are certainly nutrients that we know are really important for root growth and fruit quality, photosynthesis, things like that. Again, in this trial, we did not see any changes to overall plant growth or yield. But again, we were only able to continue this trial for about 3 years. And so we're, certainly curious as to what other changes we might see in the long term. But again, it might depend on the rootstock as to whether you're seeing some of these big changes. So like I mentioned, we have a new trial here at the Research Center. Again, my student Emma Dawson is really working on this. Again, these trees were planted in April 2023. They've all obviously been covered with IPCs, and so should not have HLB. We're planting cover crops twice a year, and then we also have on top of that a compost treatment. And so underneath the canopy of some of these trees, there is being applied a plant-based compost twice a year, and again at 12.4 tons per hectare. We don't have a lot of data on this trial yet, but we do, hot off the press, have the results of soil organic matter. We didn't see big changes in organic matter, interestingly, with the cover crops on this treatment after 1 year, but we did with the compost. And so the compost is in green, the control is in purple. And so we are seeing increases in that organic matter and the soil that is underneath that compost right next to the tree. Obviously, you can see we're starting at 0.5% soil organic matter, which is not atypical, unusual for our area. But it also means we've got a long way to go. So we're very curious to see what happens. We just sent off samples to look at the microbial community, and so hopefully we'll have that data in a couple of months. So take-home messages for compost. We've seen that compost can impact the rhizosphere, again, that area around the root, the microbes around that root based on the rootstock. And so that can make a big difference. We do see that compost can change these microorganisms around the root in a way that's related to specific root nutrients, which could in turn potentially have impacts on the tree at some point. But as I mentioned, we have a lot of questions still. We really want to see what this impact is long term on the trees. There's a lot of work that we are trying to do now looking at nutrient release and availability. And one thing that I was hoping to study, and unfortunately the project didn't get funded, but maybe we can try again, is to look at different compost types and application rates. We know that there are a lot of differences in the compost from different vendors, and we're curious as to how big of a deal that actually is, as well as that application rate. Is more always better? May not be. All right, so the last thing that I want to talk about are humic and fulvic acids, really focusing on humic acids, but fulvic acids are closely related in the general idea. And so again, this is work that Emma Dawson has been working on. And so humic acids are basically extracted from soil or organic products like lignite. And it's really an organic waste that's generated using some acid and alkaline extraction processes. The reason that they are potentially of interest is that they are extremely high in carbon, and they have a C to N ratio of around 37:1. And they can be applied either as a liquid or as a granular formula. And so they're often recommended by various groups to increase soil nutrients, build soil organic matter, and promote crop growth. However, the literature has a pretty ginormous range as far as application rates, 20 to 810 pounds per acre, which to me is a pretty big extreme. And so there are a lot of questions, I think, that are out there about what these might be doing for citrus. And so that's what we set out to do. And so this, again, was a project that was done in coordination with Dr. Uta Albrecht and Dr. Ramdas Kanasari in a commercial field where we had Valencia on US 802. These trees were planted in 2019. We used a granular humic acid, and then we had no humic acid treatments. We applied the humic acids at about a 675 pound per acre rate. Not exactly in the middle of that 20 to 800 range, but slightly on the higher side. And this project went on for 2 years, but the data I'm going to show you was collected towards the end of the project, where we collected samples right before an application of the humic acids, 2 weeks after the application, and then 6 months after the application. Because normally we are applying, or not normally, what we did for this trial is we applied them twice a year, roughly 6 months apart. And we were curious as to how quickly might there be changes to the soil microbes right after we apply these humic acids. And so that's what this data is going to show. And so when we're looking at this, we really didn't see any big changes on soil physiochemical properties with humic acids. And so right here, I've got soil organic matter. And so in the pink is the control, the green is the humic acid. Before we applied them, which is actually technically after a year and a half of application, there was still no difference in the soil organic matter with humic acids. And even then 6 months after that particular application, there was no statistical difference. POXC, or permanganate oxidizable carbon, is another metric of looking at available carbon. It's thought that this form of carbon might be more available, bioavailable to microorganisms, and so we often are looking at that. There's some slight changes, but not really what I would consider biologically relevant. They might be statistically different, but they're basically the same, and no real big changes there. Same deal with ammonium and nitrate. So we're not seeing major changes with these humic acids on these particular properties. So what about the soil microbes? Not a whole lot there either. And so here we're looking at the bacterial microbiome here before humic acids, and the control is at the same point. You can see that the amounts of some of these organisms, like say the green one, are slightly different, but in general, those are basically the same microorganisms at that time point in both. 2 weeks after, again, we see slight shifts in the abundance of certain microorganisms, but these were not statistically different. Same deal 6 months after the humic acids. So there might be a little bit of change right after 2 weeks after you apply them, but by 6 months, it's basically the same thing again. So there could be a very, very short change, but not a long-term change when we're applying these humic acids, at least to the bacteria. Some of this we also think is that this 2-week change could also be related to the time of year. We collected the first set in May, then we applied the humic acids. That's the beginning of the rainy season. All sorts of things change once it starts to rain every day. And then 6 months later, you're back in the dry season. And so any changes that we do see here may or may not actually be attributed to the humics. They could be related to all of the rain. Similar pattern when we look at fungi, again, before and in the control, more or less the same fungi in those soils. 2 weeks after, again, some slight shifts. And 6 months, again, not a whole lot of difference. I will point out one, and again, we have the same seasonal pattern. There's one change that we thought was possibly interesting. We are seeing increases in glimmeromycetes. I can never pronounce it correctly. 2 weeks after humic acids. These are potentially interesting because these are the AMF fungi, the arbuscular mycorrhizal fungi, which can potentially have benefits to tree growth. But we only see it 2 weeks after humic acids. 6 months later, they're not really making a big presence. So again, humic acid might be a short-term change, but we're not seeing major long-term changes to the soil microbiome when it's applied. So our take-home messages for humic acids is they're not really changing a whole lot of anything. And we also did not see changes to tree growth or yield. Um, there are plenty of questions, and I'm not sure that I would necessarily completely rule them out. As you saw, the application rate range is pretty big. And so maybe we're not applying enough. Maybe you need to apply them more frequently. Hard to say, but certainly the amounts that we did not seem to have much of an impact. So I want to real quickly go over one more thing, and that is related to what do we measure to try and determine soil health? I've shown you a bunch of different types of data that we collect in my program, but not all of those are useful to everybody. And certainly looking at the soil microorganisms in the way we do is not really a practical way for farmers and growers to look at things. So we have a project that's been going on where we are trying to address this exactly, in trying to figure out what are the right indicators to measure in commercial citrus groves when we're using cover crops as a sort of proxy for healthy soils. And so we were looking at these two groves that had cover crops for different lengths of time, different tree ages, and you name it, we have been measuring it. These are all the things that we have been measuring, and we also sent samples to commercial soil labs that offer a soil health test to compare our results. I will say we are still processing all of this data because we measured some of these things 3 times a year, some of these multiple soil depths once a year. It's a massive amount of data that is requiring a lot of analysis. And so hopefully I can give you guys a talk just on the results of this later this year, fingers crossed. But one of the things that we're seeing that's, I think, something to keep in mind is that it really depends on the method you use to measure a particular parameter. So this is some work being done by a graduate student in Gainesville that's on the project looking at aggregate stability, which is a physical soil indicator and not one that I often think about, but it can be very important for soil health. And there are different methods to measure this. And so she sent samples for this VAST method that I think it's called volumetric aggregate stability test. And it's a really rapid and inexpensive commercial method. However, in our trials, we didn't see any statistical differences between any of our treatments with it. However, there's another method, the Cornell rainfall infiltrometer method, where we do see statistical differences in aggregate stability between our control and our treatments with cover crops, in particular the non-legume cover crop. So what method you choose also determines whether you might see differences. And this also goes back to that soil depth question that we're trying to address as well. So how do you measure these things? What do you look at? Can sometimes make a big difference. Of course, ultimately, the big thing that everybody wants to see are changes in your yield and your overall tree production. And so that's probably the ultimate indicator. But since these are long-term processes, having some other ways of figuring out, are you on the right path, we think would be beneficial. So stay tuned for that, and hopefully I'll have some more information on that in a couple of months. So real quick, just to generally summarize things, I think soil health can be a helpful analogy to help us think about how ecosystems function. In particular, I'm really interested in thinking about how it's related to resiliency and resiliency against all of these different challenges that our citrus groves face. Certainly, microorganisms are critical components of soil health and can play a big role in your agricultural system resiliency because they are key to the formation of soil organic matter, nutrient cycling, and plant defenses. Cover crops and compost can impact soil microorganisms and soil health in citrus groves. We've seen changes in the microbes that are around citrus roots based on the application of cover crops and compost. But the impacts of these changes might be related to the rootstock, and they might be tree age dependent, and they're probably going to be dependent on the length of time that you've been using these treatments. Did not see a whole lot of impact of humic acids, and again, no impact on tree growth. But again, I think I can't emphasize enough that this is this is a long-term investment. And I know that's particularly challenging in our HLB conditions here. But these processes take time. And so changes to the trees could take a number of years. But we hope that some of the changes that we're seeing already with the soil microorganisms is an indication that bigger changes are possible. So with that, I'd like to thank my collaborators, particular special thanks to our grower collaborators, our funding sources from CRDF and the USDA, and my awesome, amazing lab team. And I'm happy to take any questions.